It's with great pleasure I get to introduce Walter Herzog, who is a professor at the University of Calgary, and many of you probably don't need to be introduced to Walter because you've probably read his work if you were involved in osteoarthritis, cartilage, muscle mechanics, cardiac mechanics, or even stroke after some chiropractic neck manipulation. You're probably aware of some of his work. So Walter is prolific in the field of biomechanics, and we're fortunate to have him here today and to present on some of his work that I think he finds dear to his heart on muscle mechanics. And so just a couple of other things about Walter. He's from Switzerland and did his undergraduate degree actually in physical education at ETH in Zurich. And before taking on a PhD in 1985, he completed that at the University of Iowa, and he studied biomechanics, also with another New Zealand connection there, the late Professor Jim Hay, and who was well known in the sports biomechanics realm. And after that, Walter did a postdoc at Calgary, and he hasn't left. He's managed to stay there the entire time, and has gone on to numerous awards, too many to review. I actually quickly pulled up his bio sketch before today, so I thought I'd say something about Walter, but it was 35 pages long, and decided probably that I'd just grab a few snippets. He's currently the director of the Human Performance Lab, and the HPL at Calgary is definitely known internationally as the premier place to study kinesiology, sports science. And he's also has joint appointments there at engineering, and you'll see veterinary medicine as well as medicine at the University of Calgary. He's a member of the Royal Society of Canada. He's got 479 publications, has been cited almost 20,000 times, so that's kind of astounding numbers. I'm going to stop there because there's too much to say, and I'll just let you take over and give us a stimulating talk on muscle. So thanks for coming, and thanks for being here.
Thanks, Tor, for a very nice introduction. It's great to be here. I've only been in New Zealand once before, in 2003 at the ISB in Otago, and then we got kind of snowed out in Otago and had to stay a couple of days in Auckland, but I didn't have time to look around. So it's really good to be here. And before I start, I knew about Tor's work, and I knew a little bit about Neil Broom and the articulate cartilage and so, but I've met a lot of interesting people today and yesterday, and students, and very, very impressed by the size and the quality of the work. And so hopefully I can match that a little bit and show you some of the stuff that we have been doing. And as Tor mentioned, there's a variety of things that we have done in musculoskeletal biomechanics, but he left it open to me what I wanted to talk about. And whenever people leave it open, then I talk about muscle contraction mechanisms, because it seems that's where my heart and soul is, and I love the other stuff too, but this is really what I like to talk about and what I like to think about. And you know, whenever I have a free moment in my research life, it's probably related that I'm trying to think about more experiments and more approaches to this thing. And since I assumed that not many people talk about or work in muscle mechanics, in the bioengineering field, I thought I'm talking a little bit about the past, just to give a very foundational level about how people think about muscle contraction, and then I talk a little bit about present observations that do not kind of fit that thinking of muscle contraction, and then talk about maybe a little bit the future, about how I think and some other people start thinking about how muscle contraction mechanism in skeletal muscles might be looking like. And so this is the outline, and so I'll be starting to talk about the past, which really is essentially textbook information about how we think about muscle contracts. And so the first thing that I always like to emphasize, and many of you will be aware of that, is that muscle, of course, has these very nice hierarchical structures. And I like to start out with the actin-myosin filament, the contractile filaments, that then are assembled in a very crystalline way in muscles, in these sarcomeres, as we call them, with the myosin filament in the middle, and then the actin filaments, bound to these connective tissue layers here that are called Z-lines, Zwischenscheibe, from a German anatomist. And then you have this in cross-section, this very nice crystalline structure of how they are arranged. And then when you have a bunch of sarcomeres in series, then you get a myofibril, And I will be talking a lot about single sarcomere and myofibrillar work, because those are very beautiful preparations where you measure force and length changes. And since the sarcomeres are in series, you know when you measure force at the end of the preparation what all the sarcomeres have to carry. And then when you have lots of myofibrils assembled, about 2,000 of them, then you get a cell with a cell membrane, multinucleated cell that is longish, of course, in skeletal muscles. And then many cells together give you a fascicle, and then many fascicles give you a whole muscle. But we'll be talking a lot about this level here, which I think is a crucial level to really get a good handle on mechanisms of contraction. We've also done some work with protein-protein interactions, but I'm not going to talk about that today at all. So, talking about the past, it's a bit confusing for people who are not in the field, because there's these two Huxleys running around, Andrew Huxley and Hugh Huxley, and for a long time when I got into this field I thought there was only one, until I realized there are two people completely not related, independently working. But magically in 1954, in the Nature issue in May, they both published work where they essentially showed one Andrew Huxley on a single fiber level and Hugh Huxley on single myofibrillar level. What they showed that under a variety of contractile conditions, this A band or myosin filaments did not appreciably shorten. And before that in the 1950s and 1940s and so, the idea about muscle contraction was that you had these A bands that upon activation somehow would undergo these helix to coil transformations and shorten, and that would produce the shortening in muscles and would produce the force. And then of course if they show no, they really don't shorten, then the question became, well, so what do we do now? And then they came up with the idea that maybe these filaments, the actin filaments and the myosin filaments, maybe they actually slide relative to one another, and you don't have shortening of a protein, but you have a sliding of proteins relative to one another. And this then was referred to later on as the sliding filament model. And then we had to figure out, or they had to figure out, well, how does this sliding occur? And so people were looking more at the structure of the myosin and actin filament. And this is kind of a conceptual structure of a myosin or thick filament, with the backbone here that's made up of this tail portion of the individual myosin proteins. And then these myosin proteins have these two heads that stick out and that we now know attach to the actin filament. And then they are very systematically and again crystalline ordered along this myosin thick filament, whereby you have from one of these structures to the next one, longitudinally you have about a 14 nanometer distance and also about a 60-degree turn. Then you get another one of another 60-degree turn and another one, and then you have them offset by 180 degrees. So once you go 3 lengths of myosin filaments head sticking out, you come to the same arrangement again. And so you really have these cross-bridge heads sticking out about every 42 or 43 nanometers in the same direction and supplying the same actin filament. And the interesting thing for me was when I got into this is that I always thought there were thousands of myosin cross bridges interacting with actin, but a half sarcomere, a half myosin filament is only about 720 nanometers long in a skeletal muscle, in a vertebrate skeletal muscle, and then every 43 nanometers you have one of these. So you really only have about 17 or 18 or 19 of these cross bridges interacting with the same actin filament. And then you have 3 myosin filaments that supply the actin, and so maximally you have about 54 cross bridges that can attach. But we know that never really happens, because estimates in an isometric contraction are that you have about 30% of the cross bridges attached at any given time maximally. So really only talking about a handful or 2 handfuls of cross bridges that interact with a given actin filament, not hundreds and thousands like I thought when I really didn't know much about this yet. So it's a very small number of sarcomeres. And then you have the actin filament, that is made up of these chains of actin globules. So you see these 2 chains that are kind of wrapped around each other like a pearl necklace, where you have 2 strands and you kind of wrap them around each other. And then in the groove of that necklace of these 2 filamentous G-actins, you have this long filamentous protein, the tropomyosin, a regulatory protein, and then every 36 to 38 nanometers, you have this tri-complex of troponin C, T, and I, and the troponin C stands for calcium, is the one that binds calcium. They're called regulatory proteins because upon activation you have calcium binding to the troponin C, and then you have this movement, this reconfiguration of the troponin-tropomyosin complex that then supposedly frees up attachment sites on the actin filament, and only then can myosin crossbridge heads attach to the actin filament and start interacting. And then one of the Huxleys, Andrew Huxley, so that's the one that won the Nobel Prize in 1963 for nerve transduction in the giant squid axon. So he then came up with a mathematical formulation of how this interaction between the myosin and the actin filament might be taking place. And so he had a very, very engineering-like model where he had these side pieces, as he calls them, that emanate from the myosin filaments, and they have then certain attachment sites on the actin filament, and then this thing, in his theory, just wiggles around by Brownian motion, and then every once in a while, when it's in the neighborhood of an actin attachment site, it has a certain probability of attaching, and the probability is given by this F function, and the probability of detachment is given by these G functions here, and these are the original functions that he described in 1957, and the important thing to realize here is, for later on, that these F and G functions are only functions, so the rate constants are only functions of x, where x is the distance of the nearest attachment site relative to the equilibrium position of the cross bridge. And so you have this stochastic interaction between actin and myosin, and then the force that is produced in a cross bridge depends on how far these elastic springs, they were assumed to be elastic, how far these elastic springs are pulled by Brownian motion out of alignment. And then you go and ask yourself, well, why do we need energy if everything's happening by Brownian motion? So of course, when you have two proteins interacting, either the binding or the unbinding requires energy. And in this particular case, the way it was formulated and the way we know it's probably correct today is that the unbinding requires the energy. So interestingly enough, most of the energy in a muscular contraction is not really in the force production, and so it's in this particular model, it's in the unbinding of the myosin from the actin filament. And if you don't have any ATP which delivers that energy, then you don't have any unbinding, and that's when you are dead and you don't have any ATP anymore after about 2 or 3 hours. Then you go into this Riger or rigor configuration, and your cross bridges essentially remain attached. And then the other Huxley, Hugh Huxley, in 1969, as part of his PhD thesis, made the observation that volume is not only observed or contained and conserved in a whole muscle, as people had known, but it's also conserved within a myofibril and within a sarcomere. And therefore, when you have a short sarcomere, the distance, then it gets wider, and therefore the distance between the actin and myosin filaments get larger. And then he was saying, in this previous model here, it's really hard to imagine that this distance can change quite a bit and you can make interactions with this type of a setup. And so he said, we need something where this cross-bridge head, this side piece, can move away from the backbone of the myosin filament, and therefore had a joint here. But then he also argued that this configuration, then if there was not another joint, then this configuration would not really be stereotypically attaching here, but would also always be in a different configuration. So he introduced a second joint so that the head could always attach in a similar configuration, independent of how far the actin-myosin filaments were apart, and then came up with this idea that the cross-bridge head might be turning. And then the other Huxley, Andrew Huxley and Bob Simmons, in 1971 came up with this very nice formulation of the cross-bridge theory, where you now have multiple attached states and also multiple detached states. And so now all of a sudden we have a multi-state model for the interaction of these two filaments that produce force. And then in 1993, even Raymond developed an atomic structure of the actin filament shown here, and the myosin cross-bridge head here. and then based on that, a proposal for really how the mechanical interaction and the biochemical splitting of the ATP that provides the energy for this muscular interaction, for this actin-myosin interaction, how that works was then given. And so we have then a cross-bridge model that is driven by chemical processes and also by mechanical different states of the mechanical states. And the mechanical states influence the chemical states, and the chemical states influence the mechanical states. And so it's a very interesting interaction that happens. And then for those of you who do some modeling, and we have done some cross-bridge modeling, the way most people then think about cross-bridge models is that you have these mechanical or biochemical states, very distinct states. Here, for example, 5, 2 detached ones and 3 attached ones, and they're governed by these backward and forward thermodynamically consistent rate constants that are given from state to state, and that are only dependent on this x distance that I was talking about before, this distance of the equilibrium position of the cross bridge relative to the nearest attachment site. And then that's a relatively simple stochastic system where you then can start doing some cross-bridge modeling. So the assumptions that Huxley made in 1957 for that very initial basic model was that, every cross-bridge produces about the same amount of force, so there's not strong and weak cross-bridges. That cross-bridges act independently. We are fairly certain today that that's actually not correct. But for the mathematical formulation, it was easier to do that. But a lot of cross-bridge models now have an interaction between cross-bridges. It's fairly certain that when a cross-bridge is attached, neighboring cross-bridges are more likely to attach than cross-bridges that are farther away. We use 1 ATP per cross-bridge cycle, and for most normal interactions between actin and myosin, I think people still believe that. And then, of course, we have a uniform arrangement of cross-bridges along the myosin filament, and uniform arrangements of these attachment sites on the actin filament. And then when you have these few assumptions, then you can start doing some modeling with this. And so this model, this cross-bridge model that initially was developed in 1957, has been really good for predicting the forces and stiffness and the energetics in isometric contractions. That means when a muscle or a myofiber or a fascicle are held at a constant length, or when you have a concentric contraction. That means when the muscle is shortening while it is activated, and the muscle, of course, tends to shorten when it's activated. However, and Huxley was very open about it in 1957 already, that for eccentric contractions, a lot of things don't fit. And I always like to emphasize that Huxley in 1957 already knew about that and wrote extensively about it in that enormous paper that he wrote. The force, the eccentric force was much too high, the energy requirements were way too high, the stiffness didn't add up. And so he knew that for eccentric contractions, that means when you have a muscle that produces force and is activated, but you apply to the muscle a bigger force than the force of the muscle, then you pull the muscle to longer lengths. That's an eccentric contraction. And he knew already then that something's going on there that is really not well predicted, and but really didn't have a solution. And so in 1980, if you're ever interested in muscle mechanics, Andrew Huxley wrote this fabulous book, Reflections on Muscles, and the whole book is full of little treasures. And so I'm showing you some here on muscle stretching or eccentric contraction. So in 1980, he said, that I imagine that special features have evolved which allow elongation to take place without damaging the muscle. And I'm going to be talking about what I think that special feature might have been that was discovered in 1976 by Murayama and then rediscovered by Wang in 1979. So there's certain structures that were discovered just a couple of years before he made that statement, but nobody really knew what these structures might be doing. And he also felt that there was a very exciting field of investigation there that might hold a number of surprises, again, on muscle stretching, on eccentric contractions. And then there was a statement in there that I think is particularly important for the talk that I'm giving here today. He said, there are several reports that a very large stretch applied during the plateau of a tetanus leaves the tension raised for a long period. That doesn't mean anything to you, but I'm going to show you what that means, because this refers to a muscle property that had been known at least since the 1940s, and had been consistently observed, but he just couldn't predict it with the cross-bridge model. And that's where I'm coming to kind of present observation, although it's not really present observation. These are observations that have been made as much as half a century ago, but really became more relevant in the last 10 or 15 years. And the observation that I want to talk about a little bit more in detail is that long plateau, that elevated force, that elevated tension that Huxley is referring to in his 1980 booklet there. So what that meant is, imagine you have a muscle or a fiber, and you keep it at a constant length. So length as a function of time is constant. You just activate and deactivate, and you get a certain amount of relatively steady force, a little bit sag there because the muscle gets fatigued. And now you take that muscle or fiber at a slightly shorter length and you stretch it actively, then you get this little bump that is very well explained in the cross-bridge theory. But then you get a higher force here, and if you stretch the muscle a bit longer, you get an even higher force. And when you shorten the muscle to the same length, you get less force. And so the question then became, what's happening here? Because we are at an isometric contraction, the same activation, the same lengths, and according to the cross-bridge theory, you should get exactly the same force. But you can easily get only half the force, or easily, in some instances, we can easily get twice as much force as is predicted. And so the question then becomes, well, how is that? Because in the cross-bridge model, if you pull a muscle, then you get a transient increase in force, but then as it approaches steady state, the transient disappears, and the force should be the same at steady state as the isometric reference force. But that's clearly not the case, and there's hundreds of articles out there that have shown that, and I'm only aware of one person who pulled a muscle and tried to find that property and didn't find it, and that was Jerry Loeb and Steve Brown on the caudalis muscle of the cat, and then we repeated that experiment and we found it in that muscle as well, and I'm not really sure why they didn't find it. But anybody else who seems to do this type of experiments seems to find this enhanced force after a stretch. So for a long time, for a very, very long time, nobody really bothered about this, because the argument was, we can explain that on the muscle or the fiber level with the structural rearrangement of sarcomeres, which is referred to as the sarcomere length non-uniformity theory. So what people were arguing for a long time is that this particular part here, the descending limb of the force-length relationship, so this is the force that a sarcomere can exert as a function of its length. And here you have this negative slope. And so people were arguing on this negative slope there's instabilities, and if you're not very, very careful, you get gross non-uniformities of sarcomeres, and we can explain this force-enhanced state with these non-uniformities. And the argument went as follows, that if you have an isometric contraction and you're very, very careful, then you have relatively uniform sarcomeres, and based on actin-myosin filament overlap, you get this force. If you now deactivate the muscle, pull it to a little bit longer lengths, and then reactivate it again, then again you have a set of relatively uniform sarcomeres, and your force is a little bit lower because now you've lost more actin-myosin filament overlap. However, the argument went that if you now have this muscle at the short lengths and pull it to this long lengths, that you would induce these instabilities, and sarcomeres would then become non-uniform, and some would maybe stay here, or in the ideal case they would shorten a tiny bit and would go here, whereas since the average sarcomere length is now this long, you also would need some sarcomeres that are much, much longer than actin-myosin filament overlap, and so they would not be exerting active force anymore, but only passive force. They would be hanging here on the fibrous elements of the muscle, and then when you add up the forces here and add up here, then you get an equilibrium, and this force is much higher with these two sets of non-uniform sarcomere blocks here compared to if all sarcomeres were at the same length. And I never forget when I read that the first time, this whole instability business, and that was when I was a student. I hadn't done any muscle mechanics at all yet. I hardly knew what a muscle was, and what bugged me at that time already was, purely intuitively, that I'm going, why would nature evolve a molecular motor that is consistent across all species as far as I know, and about 60% of its working range would be unstable, and you tear yourself apart, that you put some sarcomeres into these helpless situations where there really is no active force anymore. And somehow that didn't make any sense anymore. And I guess later on, about 10 or 15 years later, when I started doing work more on the molecular and cellular level, we came back to that. But anyway, that was the theory, and that was the explanation that still today most people would argue is the right thing. And so I have to emphasize that when you talk to the average muscle physiologist, most people will explain force enhancement in this particular manner. However, there were some people, there's about 4 or 5 models out there on the sarcomere length non-uniformity theory, and it allows you to make very nice predictions. One of the predictions is that since the active sarcomeres cannot be in a more favorable position than here on the plateau, in the enhanced state you should never get forces that are up here above the plateau. When you do some dynamic modeling in a dynamic equilibrium, Ken Campbell in Kentucky gets actually slightly higher forces, but we talk about 1% or 2. You cannot really get substantially higher forces. So force in the enhanced state cannot exceed the isometric plateau forces. And then we and other people did some experiments and in single fibers here, for example, and also in other preparations, and found fairly quickly that no, you can easily get on average 10 or 15% more force. And you know, and some of these ones that have the standard error here, some of these were as high as 25 or 30% higher than the plateau. So here we have the isometric forces, and here the steady state forces after a stretch. And so we showed that, and then other people have shown that as well. And so we go, well, maybe there's something else that plays a role here. And I should go back here again. And then I presented this a while ago, maybe about 8 years ago at Woods Hall, and people were arguing, oh no, you know, that this could be whatever, you know, things. And I remember I was kind of in desperation. I kind of yelled out to the public there. I said, well, so what do we have to do that you would believe us that you don't need sarcomere length non-uniformity? And Jim Spurich stood up and said, well, do the thing on a single sarcomere. Because if you have one sarcomere, then you can't have non-uniformities. And if you do it in one sarcomere, you get the same properties, then it's happening. And so then we started doing experiments. So this is a single myofibril with 1, 2, 3, 4 A bands. And usually we did experiments in this particular way with the silicon nitride lever here. And that measures force and the motor here. And then we just introduced a third motor where we go in here now. And just to give you a little bit of dimension, this whole thing is about 10 microns long, so that's about 1/10 of the thickness of a hair, and the thickness of a myofibril, the very elastic myofibril, is about half a micron, so that's about 1/200 of the thickness of your hair. And so then we isolated mechanically a single sarcomere and for the first time, and then did exactly those type of experiments on a single sarcomere. And here you see one of those results. So this is a relatively long sarcomere length where we get little force because we are at about 3.4 or 3.5 microns, so very much down the descending limb. And then we pulled that from optimal length, so we have relatively high force, and then pulled, and now the force should be dropping down here, but it stays much higher than the steady state force, despite the fact that we are now also at this very, very long length. And not only that, this is the plateau force, so the optimal overlap between actin and myosin. We also get a higher force, and we did that on up to about 20 single sarcomeres. And Tim Leonard did these experiments in my lab. He was a PhD student at the time, and found that when you do isometric contractions, you know, you get about the force that you would expect theoretically. When you do the isometric contractions here and normalize to 1.0, it falls nicely into place. But then if you stretch it and look at the steady state forces, you get that enhanced force, and you get that force above the plateau. And so we felt fairly certain that, well, we can do this in a single sarcomere, therefore there's no non-uniformities. But then people started saying, well, you have 2 half sarcomeres, and therefore they could be non-uniform. But of course, you're all engineers. You're going, okay, that argument doesn't count because the 2 sarcomeres are not independent. If one is in a certain state, then it's predicted what the other one has to be. and if you then predict what the passive force would have to be in that other half sarcomere to always match the active force of the active sarcomere, you get a very, very strange curve, and you go, that's just not going to be possible. And we published that as well, but nobody seems to pay any attention to that. So that's kind of the fun thing. That there's other predictions that you can make. The other prediction is that this limb here, where the force increases with length, was based on potential energy level is stable on the mathematics that governs the sarcomere length non-uniformity theory, and so theoretically you shouldn't see force enhancement here, but you do, and we have seen that in other people as well. The other thing is, another prediction of course, is that in the isometric contractions your sarcomere length should be about uniform, but they're not. In fact, in isometric contractions, the non-uniformity in the sarcomeres on the myofibrillar level and the fiber level is about the same after stretch. And the other prediction, of course, would be that you should have two very distinct sarcomere lengths, but what you really get after stretch, or also in isometric contraction, you get sarcomeres at all kinds of different lengths randomly distributed. And so there's not really these two camps that then do that. And of course, those of you who are a little bit into muscle physiology will say, how can that be that I have, this much overlap, this much overlap, this much overlap, this much overlap? So I have all these non-uniform sarcomeres, and they are at steady state. They are not moving anymore. And I think one of the solutions might be that we think about sarcomeres in the wrong way. I think sarcomeres are not independent contractile units. I think sarcomeres are somehow through the Z bands connected, and therefore there is not really a wall of a Z band where you have a force on the one side and a force on the other side that has to match. But the sarcomeres are connected, and therefore what the individual overlap of a sarcomere is, the myofilament overlap, is probably less important than we have thought for a long time. But again, there's a lot of work to really show that, whether or not that's correct. So anyway, we came to the conclusion that sarcomere length non-uniformity is likely not the dominant factor or an important factor in this property. Then the question became, well, if we throw that one out of the window, what else could it be? And that's where we kind of come to the new ideas and the new models. And the new model really, for me at least, started in 2002, where I did experiments on cat soleus. This is a cat soleus muscle that I did together with Tim Leonard, and I did these experiments myself. All these experiments that other people did on single fiber level or sarcomere level, I always did them first on cat soleus because it's a beautiful muscle and easy to do. And so we have a lot of data there. But what we tried to do here was a completely independent experiment. We wanted to look at certain properties in a highly stretched muscle, and you see that it's highly stretched because before activation you have quite a bit of passive force. And so we just did some passive stretching, and then we got about the same passive force, and then we did some active stretching. We wanted to look at force enhancement, and then we deactivated the muscle here, and then all of a sudden we deactivated it, and there's it's at the same length, and there's no activation anymore, and we have more passive force. And I remember Rachel Shacker, a master's degree student of mine, was there as well, and she was on the computer regulating the length changes, and I was on the muscle making sure that the muscle was nicely supplied with fluid and didn't dry out and all those type of things. And I said to Rachel, you're pulling the muscle too long, you don't relax it. And she said, no, it's exactly the same length. And I said, no, I see the passive force is way too high. And she said, no, I'm doing the right thing. I said, get away, I'm going to do that. So I went to the computer and I did the things. And then I realized that no, when we are at these long lengths, the passive force here persists for a very long time, minutes and minutes. That's higher at exactly the same lengths when you pull the muscle actively. And we called that then the passive force enhancement, and published that in Journal of Experimental Biology. And that was the first mention of an enhanced force in the passive state after an active stretch. And the interesting thing is this stays there for minutes and minutes, but if you just shorten the muscle to its initial length and go right back within half a second, all the passive force enhancement is gone. So it looks like something has hooked in, and if you relax the muscle and go back, then you unhook it somehow. And so that was very intriguing to us, and so we thought, well, maybe this passive type of thing might have also to do with what we see during the active muscle. And then we did that on a variety of different structural levels, And probably the most important structural level was then a single myofibril, where we activated a single myofibril, stretched it, and then deactivated it and looked what the passive force was. And then we did it passively and looked what the passive force was at a relatively long length, and then did that in about 20 myofibrils and found that there seems to be a higher passive force also there. And the reason why the single myofibril is so important is because in a single myofibril, all structural proteins are gone, and really the only structural protein that's left that provides passive force is titin. And then we did some other experiments. These are again from Tim Leonard from his PhD thesis in 2010, where we said, okay, let's now take single myofibrils and sarcomere and pull them to very, very long lengths. And the lengths that are beyond actin and myosin filament overlap, which in a rabbit psoas is about 4 microns. So then here we know there's no active force anymore possible because the actin and myosin don't overlap anymore. So you pull them passively, and then we activated it here, and then the question became, here when we get into this zone where no active force is possible anymore, do we fall down, or is there really an engagement of a passive type of an element that then gives us a higher force here? And the solution was very convincing. You go beyond actin-myosin filament overlap, and at about 4.5 or 4.8 microns, all sarcomeres and all half sarcomeres are visibly and measurable and quantifiably beyond actin-myosin filament overlap. And then you go, wow, what is this? Where does this come from? And you then eliminate titin either actively or passively, and then, as I said, actively or passively, you don't get any contraction, you don't get any force in a myofibrillar setup. So a sarcomere or a myofibril needs titin for the transfer of forces from the actin to the myosin to the Z line, and if you don't have titin, nothing's happening anymore. So we're fairly certain that maybe that titin might be responsible for this difference here, and this is about a 2.5 or 3 times increase in force of a passive force in an activated state compared to a passive force in a passive state. So now you're sitting there, those of you who don't work with muscle, say, what the hell is this titin thing, So you know by now that you have this myosin filament in the middle of the sarcomere, and then the actin that goes into the Z line of the sarcomere. And then you have titin that starts here at the Z line, is anchored in the Z line, also for the first 50 nanometers about, has attachment with the actin, so it is bound to actin here, but then here it runs freely, and it is a spring-like protein, so you can pull it out to very, very long lengths, and when you release it, it just kind of folds up again. And in a certain range, it's highly elastic, and when you then get to unfold certain elements, they are called immunoglobulin domains. When you start to unfold them, then all of a sudden it becomes very viscous, a very viscoelastic behavior. But anyway, you have a molecular spring here that kind of keeps the myosin nicely aligned in the center of the sarcomere, and then it's here rigidly bound to the myosin filament and ends here in the middle of the myosin filament. And then you have another one oriented 180 degrees opposite that goes to the other half of the sarcomere. And so the idea was that maybe something happens to this molecular spring here, and in fact, the hypothesis at the time was that maybe titin is a calcium or activation dependent molecular spring. Maybe when we activate a muscle and calcium is flooded into the system, maybe something happens to the stiffness of titin, and when you then pull the sarcomere, you get more force in this structural protein than you would when you stretch it passively, and there's not much calcium there. And so for about, we had then the following speculation for the past 10 or 15 years, try to identify some of these points. So the speculation really was that you have titin here bound to the myosin, and then here to the actin and the Z line, and you just stretch the thing passively, and everything stretches out nicely, and you get a certain amount of passive force. And then the idea was when you activate a muscle, then 2 things might be happening, we felt. The first one is the calcium might be binding to certain places on this protein and reinforce some of the molecular bonds, and therefore it would be harder to pull apart and essentially would make it stiffer. So that was one way that we thought this could happen, that you make the spring inherently stiffer. And the other thing that we could think about is that since titin is already bound at the very, very end here to actin, we thought, well, maybe it can bind along actin further upon activation, and maybe therefore you bind part of your spring and it's not extensible anymore because actin is essentially rigid, and then you only have a small part, this distal segment here, that elongates, so you have a smaller spring that then is elongated more and therefore would also be stiffer because it's smaller. And not only that, these distal segments are known to be quite a bit stiffer than the proximal ones, so not only do you make the spring shorter, but you only leave the stiff elements and therefore would make it even doubly stiff, and then you get even a higher force. And so we try to identify these two things. So the first thing we did was a very simple experiment. We took a myofibril and we said, well, we just stretch this single myofibril at different calcium concentrations. The only problem is when you throw calcium into the soup there, then you start having actin-myosin interactions. But there's a variety of ways of inhibit that. And you can do it with certain chemicals, BDM, butanoidine monoxide. In this particular experiment that I'm showing you is we eliminated the troponin C, so there was no calcium binding possibility anymore, and therefore this attachment site was never really freed up for attachment. Then we did some mechanical experiment and showed that before troponin C depletion you get force at different sarcomere lengths, and afterwards this brown trace here you don't get any force. We did gel electrophoresis to make sure that troponin C was really eliminated and verified that yes it was. And then we started stretching these single myofibrils at a resting calcium level, and then at an activation calcium level, fully activated muscle calcium level, and just looked what the force was. And indeed we found that when you stretch to the same lengths in the high calcium, at the end of the stretch you have a higher force, and that force seems to persist for a longer period of time. So it appears that by eliminating actin-myosin interactions, yes, we made this spring somehow stiffer. And then the question became, well, how might that happen? And so we then recombinantly took several elements of the titin and tried to see where might calcium be binding. So in this particular case that I'm showing you here today is we took a proximal immunoglobulin domain and recombinantly reproduced it with a plasmid as what was given to us by Jane Clark, who did a lot of that work out of Cambridge in the UK. And then we used fluorescence spectroscopy to see whether or not binding of calcium occurred in the immunoglobulin domains and other domains that we tested as well. And then did atomic force microscopy to see whether the calcium binding made a mechanical difference. And just to go through this quickly, yes, when we had the fluorescence intensity measurement, this is the blank immunoglobulin domain 27, we add a little calcium, we add more calcium, we get a depression indicating that there's binding. We take the calcium out, it goes back to resting levels, so we have a nice dose-dependent calcium binding, it appears. And then we took these 8 immunoglobulin domains and used atomic force microscopy and grabbed them wherever you grab them, by accident. And then you pull them, and then you get this sawtooth type of a pattern whenever you have an immunoglobulin domain unfolding. And we did that in a resting and in an activation calcium solution. So this is with the resting calcium, this is with the activating calcium. And then since this is very, very noisy, if you have done atomic force microscopy, you know this is very noisy. So you do hundreds of experiments, and then after hundreds and hundreds of experiments, you see that in the calcium activated ones, it takes more force to pull these immunoglobulin domains apart than in the low calcium. So had fairly direct evidence that yes, calcium binds, and yes, it makes mechanically a difference. And Siegfried Labeit and Henk Gronzier already had shown before that the glutamate-rich region binds calcium, and that makes this PEVK area of titin, which is just an area of titin, makes it stiffer. And then Venus Juma in my lab showed the same thing, and then I just showed you the immunoglobulin domains that was done by Mike Duval as part of his PhD thesis. And now a lot of people have done a lot of work on titin, and we know now there's phosphorylation sites, there's more calcium binding sites, there's other structures that can bind to titin, and they all seem to have a mechanical effect on this thing. And so I'm only showing you a very small part. Really the king of this particular research is Julio Fernandez. He knows all the 20,000 things that can bind to titin and all the effects they have. And for us, the calcium was particularly important. That's what we wanted to look at. So then the next question was, okay, so we know the calcium binds, we know it makes it stiffer in certain elements, you know, these immunoglobulin domains that we looked at, and then the PEVK region that we and also Henk Ronzi and Siegfried Labeit looked at. So the next question became, does it also bind to the actin and shorten its spring length? And for a variety of reasons, we thought that this very unique N2A sequence here might potentially be the key for binding, that this might actually bind the whole proximal part. And so we did a mouse deletion model where we deleted a big part of this N2A region, and then did the same experiment over again where we stretch this mouse psoas muscle passively for a wild type mouse and for this MDM mouse that has part of this N2A region missing, and the passive ones are indistinguishable. And then for the active wild type, we again, when we go beyond 4 microns here, we get this vast increase in force compared to the passive that we also saw in the rabbit psoas. So we see now the same in the mouse psoas as well. But now we also stretched it actively in this mouse where the N2A region was missing. And the question was, is it now going up here or is it down here? And what we found was something a little bit in between. We got a little bit higher force, but clearly not up here. And so the interpretation was that maybe we have some calcium binding here that makes the spring a bit stiffer, but maybe we don't have that shortening of the spring because the N2A region was missing. But there's a lot of problems with this particular thing, because when you do deletions like that, you don't know what happens to other proteins, and our initial force here was significantly smaller at plateau, at full overlap, compared to here. So we really don't trust these results very much. They are published, they're out there, but with many limitations added at the end, so that if somebody reads it, hopefully they will not just say, oh wow, the N2A region is very responsible for all that. There's a lot of caveats there. And so we figured we need to do a little bit of cleverer experiment. And so what we try to do now is to see whether or not we can see if this proximal element here might get stuck and doesn't elongate when we actively stretch a myofibril. So we did some antibody labeling of titin. So we had antibodies that we tagged with a fluorescent label, and so we then divided the titin essentially into a proximal and a distal segment at the PEVK area, and then did really a simple experiment. Mike Duval, it took him 3 years to get this all going, but at the end it's a very simple experiment. It's very hard to do technically, but it's very simple conceptually, because you have a label here. I'm here showing several labels, but at the end there we only used this label for the experiment that I'm showing you at the end of the PEVK region. And the idea was, well, if we stretch this thing passively, then this proximal segment and the distal segment should be elongating according to their spring properties, whereas if we actively stretch it and some of the proximal part really binds, then we should see very little or no elongation of the proximal part, and then the distal part will have to compensate for that. So conceptually, a very simple experiment. And then my student, Mike Duval, again did this. And so what you see here is the titin segment lengths, and here you see the 2 titin segments, and this one is the one of importance because that's the distance from the Z line to this marker. And so we measure this distance as we stretch the myofibril and the sarcomere. So here we stretch the half sarcomere and the sarcomere, and then you see that this distance is increasing in the passive state, so exactly like we expected. The thing should elongate, everything should be elongating. However, if you then did this actively, and I'm only showing 2 myofibrils, Mike has done this in over 120 myofibrils, and they essentially show the same results. So what they show very consistently is that when you now measure this distance in the active myofibril, it appears that for a little bit there seems to be some elongation that's very similar to the passive one, but then all of a sudden it doesn't elongate anymore. And here again it's elongating, and then here at about 2.7 or 2.8 microns it's not elongating anymore. And so the idea is, initially, we speculated this would not elongate at all, because upon activation the thing would bind to actin. But what seems to happen is that you stretch a little bit, and then it might get stuck here or bind to actin, and then you don't elongate this part anymore, and then the whole elongation has to be taken up by the distal part. There is a variety of different interpretations of this result, and this is how I'm interpreting it. Interestingly, Mike Duval, when he defended his thesis in the paper he's writing right now, he actually interprets these results very, very differently, which is very interesting. So we have kind of a 180-degree, we look at the same data, and I love this interpretation, he loves the other one. And I'm not going to talk about the other one. but you know, it's one of those funny situations where two people look at exactly the same data and come up with a different theory. But I personally, and the reason I'm presenting this one, it's not because it fits better, but it's a very simple explanation, and I also find it a very beautiful explanation. But of course, it might be incorrect. But what we do know for certain, that's where Mike and I agree, is that in active and passive stretching, the proximal segment does not elongate as much. We just differ in why that is. Now, I like the binding to actin type of thing. But there's clearly something going on that more of the stretch has to be taken up only by the distal segment, and therefore you stretch it more, and that will produce more force. Therefore, we agree. And here is just more of the same results of multiple myofibrils. So I'm going to come to the conclusion here. So what I tried to show today is that titin is a very soft, passive, very soft spring in passive stretching. And I think that makes a lot of sense, because when you move your arms or legs, you don't want to have a lot of passive resistance when you do that. Children with cerebral palsy or people with strokes have difficulties, moving their limbs through the full range of motion because of some passive resistance. You don't want that in everyday regular movement. But titin seems to be getting stiff in active muscle, and so we think that's partly due to calcium binding. And I think most of the people who work in this area would agree that calcium binding occurs, and calcium binding will affect the passive property of titin. I think most people in this area would agree with that. We also think that titin might be binding to actin. That's clearly not established yet, and there's a lot of people who would argue that that's probably not the case, and some argue that is the case, but there's a lot more work that needs to be done there in order to really verify that. So really the new mechanism that I'm kind of envisioning and how this passive force enhancement, the active force enhancement might be explained, is that a muscle, when you passively stretch it, has a certain amount of regular passive force, and what might be happening, what we think is happening, what we see in myofibrils happening, is that upon activation, the slack lengths, where you start seeing titin force in the myofibril, is at a shorter sarcomere length, and the stiffness is higher, so the force increases faster. And so we think that you have really a shift in this passive structural element, in this force contribution, when you do that. So I guess as a take-home message, we think that force regulation might not only happen in actin-myosin, but that some structural proteins, particularly titin, might potentially affect, I'm not really sure what I should say, active force or passive force. Seems to affect force in one way or another when the muscle is activated, and titin does appear to play a certain role here that at the moment is essentially neglected, except for 2-3 people who have incorporated that type of thinking in theoretical models, but it's really not happening on a bigger level, and there's a variety of good reasons probably for that. And so titin helps when actin-myosin is weak. I think it's very nice. You lose overlap. The machinery, the contractile machinery doesn't work well anymore. Titin comes in. And the other thing that's really neat about titin is that, of course, when you bind between actin and myosin, you don't have to bind and unbind, like cyclically, like actin and myosin. So it's energetically very efficient. And the interesting thing is that in some non-vertebrate muscles, there's a titin analog called twitchin. And twitchin is a protein that, for example, in mollusk muscles, can exert a very high force for hours and hours with virtually no energetic cost. And what it does, what twitchin seems to be doing, is that upon activation, it anchors itself between the actin and myosin and binds very rigidly to it in a shortened position, and then retains an enormous amount of force. But there's no cycling there, so there's really, you just anchor it in once, and then you get force for hours and hours at little cost. And twitchin is very, very similar in its structural arrangement and also in its structure, how it's built, to titin. So we think that titin might have a shorter living but very similar role to what is observed in non-vertebrate muscles, in some non-vertebrate muscles. So I want to quickly finish up. This is Andrew Huxley, 1999, came and visited Calgary and spent a couple of weeks with him. That was kind of the most exciting 2 weeks in my life, and I would like to repeat what he says in his 1980 book there. I imagine that special features have evolved which allow elongation to take place without damaging the muscle. I think some of these special features might be the titin that was essentially unknown in 1980. And of course this shows Andrew Huxley with my 2 boys, 6 and 8 years old in 1999. He wanted to go to the dinosaur Drumheller Museum, and in 2019 we organized the International Society of Biomechanics conference again in 2019, and you can go to watch the Drumheller Museum there. Andrew Huxley was there, so you should go as well. Thanks a lot.
Thanks, Walter. It was a great talk. We have time for some questions. Dennis.
You've just given us a wonderful presentation on skeletal muscles, so I'm going to take the liberty of asking you something about cardiac muscle.
In this particular property, there is one abstract from us out there that says, presented to the Biophysical Society 3 years ago, that says exactly the same property essentially exists in cardiac muscle. And then there's a paper out there by my former postdoc, Dilson Rassier, who's now at McGill University, and he says force enhancement does not exist in skeletal muscle. So there's really 2 written accounts out there on this particular property, and with 2 completely different results. I would argue it's there because we have seen it. And if you do look at the Rassier paper, it's very, very interesting because he shows a single sample trace of cardiac muscle, and it shows an enormous amount of passive force enhancement, but no force enhancement at all. And during the stretch, there is a very quick increase in force, and then force drops almost down to 0. And he pulls it very, very fast, at 10 microns per second per half sarcomere. And I think what he did is that the muscle was slipping. And when we do that in cat soleus, when we pull it very, very quickly, then we don't get the force enhancement, we get exactly the slippage response. So I think actually his results are not correct for normal physiological stretch magnitude and stretch speeds. So I think it does exist. That would be my hunch.
But it's a shorter molecule, is that right?
It's very interesting. Titin is much shorter in cardiac muscle, and in rabbit psoas, for example, we need to go to about 2.7 or 2.8 micron sarcomere lengths before we see passive force. In cardiac muscle, that happens at about 1.92 microns, because the isoform of titin, I didn't even talk about that, but every muscle seems to have its own isoforms, and depending on that, passive force comes in at shorter sarcomere lengths or longer one. And cardiac muscle, to my knowledge, is by far the shortest one where titin starts playing a role at 1.8, 1.9, 2 microns, and very, very stiff. Because when you get to the end of the plateau in cardiac muscle,
you don't want to go any further.
your titin provides as much or more force than the active muscle can provide. It's a very, very stiff preparation.
Paul.
So it worries me in the case of passive stretching of the titin, your explanation of these globular structures sort of unraveling and creating the sawtooth. Like force-length relationship. Doesn't that go counter to the argument that titin is trying to, or acting to center the myosin, because there are going to be multiple positions where the myosin can sit. That satisfies the force.
Well, 2 things to that. The one is that we do know that very often the myosin is not nicely centered in the center of the sarcomere. It actually does go back and forth, and especially at shorter lengths when titin seems to be relatively slack. Horowitz showed very, very nicely that you get more differences and more non-uniformities in half sarcomere lengths. At longer lengths, that doesn't seem to be happening as much. And then the second thing about that is that, yes, these immunoglobulin domains, they unfold and then they have this sawtooth pattern, but there's a big there's a big controversy between the big people in this area whether or not immunoglobulin unfolding is actually physiologically important or not. So the two big guys, Wolfgang Linke would say yes, it plays physiologically a role, and immunoglobulin domains start unfolding is in the normal range of movement. Henk Ronzi would argue that's not going to happen. And when you pull in rabbit psoas, you start having unfolding of immunoglobulin domains at about 3.5 microns. And that's more than what the rabbit psoas would ever achieve. And below that, your titin is virtually elastic. And when you start the unfolding, then you get that very, very viscous response where you have an enormous amount of, in fact, you lose virtually all energy that you put in the system. You lose it all. And then there's a couple of very interesting arguments why that would be. We would argue that in the passive state, immunoglobulin unfolding probably doesn't play a role because it's not the titin is not stretched enough. But in the active state, if we are right, and a big part of the element, a big part of the protein is bound to actin, it essentially only leaves the PVK and the distal immunoglobulin domain. And so in the active state, we have argued that you probably have immunoglobulin domain unfolding. But nobody has ever seen it, and so our evidence and other people's evidence is all somewhat indirect. So there's a big debate there, and I don't think a real solution yet.
Really lovely talk. I just want to come back to that final statement that you made, which I'm sure I like. I mean, the sense that the titin's role here is to act as a protective mechanism in terms of where your sarcomere length is overextended. What experiments would you do to actually demonstrate that's the case? I mean, I guess you could look for differing degrees of sarcomere length and heterogeneity. I mean, how would you establish that it is actually adding safety there directly in an experimental sense?
You mean showing directly that it binds to the titin?
No, just simply if you disable the effects of titin, which you can do.
Yeah.
What would you be looking for to demonstrate that the effect of that is actually to reduce the stability of the structure, or make it more risk? Yeah.
So when you take all the titin out with a maltrypsin treatment, when you do that and you activate, you immediately upon activation lose your entire structure. You don't see any striation patterns anymore. So we have tried to take part of the titin out, and so that we digest about 40%. We try to get about 40% out, and if you do that, then you get more sarcomere and half sarcomere non-uniformities.
So there's non-uniformity you should measure.
So the non-uniformity, we have measured that, and it becomes bigger when you have less titin. There's a very interesting article out there that says that looked at atrophied muscle. And in that particular article, we have not reproduced that, so I have no idea whether or not it's correct, but the finding that they seem to have is very interesting. They say when you have severe atrophy in a muscle, one of the things they observed is that you have much less titin protein relative to myosin. And normally per half myosin you have 6 titins, and we know that. But it seems they only have about enough protein in this atrophied muscle for about 2 or 3 titins. So it would be very interesting to use myofibrils of an atrophied muscle where you have verified beforehand that you really lost about 2, 3, 4 titins per half myosin, and then see whether or not an atrophied muscle is also more non-uniform in the half sarcomere lengths. I would like that much better than the digestion approach, because with the digestion approach you never really know exactly what you have. Yeah. But we have used, as a measure of that, we have used non-uniformity when we took some of the titin out. Yeah.
Last one, Jeff.
Nice talk, Walter.
Thanks.
I think this is really interesting stuff. So on the 2 hypotheses for titin as an activatable spring, so the binding site or the binding to actin, are those mutually exclusive hypotheses? And what's the sense that in different orientations you might have one or the other, and that would give you different passive force profiles?
I would think that it is both mechanisms together. So that's my, so I think, I'm fairly certain calcium is bound to titin. I'm fairly certain the mechanical properties are changed, and I think most people in this area would agree with me with that. But when we do experiments, and I haven't shown these, but we also have done experiments where we pull beyond actin-myosin filament overlap, where we inhibit the cross-bridge binding and the activation. So where we allow calcium in there. And when we do that, then we get about 10 or 20% of that force differential accounted for. So I think the calcium binding to titin makes the spring only stiffer to a relatively small degree, and 80 or 90% of that force differential that we had is not explained. So there's something that very dramatic that must happening, and it seems that, and you can't do that with calcium. So I don't think the binding of titin to actin is calcium regulated, but you actually have to have cross bridges attaching, and if you inhibit some cross bridge attachment but not all of it, then you get a partial effect. So my interpretation of that is that maybe when the regulatory protein bind calcium and then the cross bridge goes into the weakly bound state and then into a strongly bound state, we know from other people's work that when it goes into the strongly bound state, there is more movement of the regulatory proteins. And my favorite hypothesis is that additional movement when the cross bridge goes into the strongly bound state, that additional movement might free up binding sites for titin. So when you have that strongly bound cross bridge, and the more you have, then you free up potential binding sites, because you don't get it. We also did it with these experiments where we didn't inhibit troponin C, but we used a BDM, a butanidine monoxide inhibitor, and that inhibitor allows for weakly bound cross bridges, and we don't get the big force enhancement, we just get the very small one. And so the suggestion is that you do need force, strongly bound cross bridges before that potential binding might be happening. But that's going to keep us busy for the next 10 years to really show that and prove that. Because conceptually these experiments are all very simple, but technically they are a nightmare. So.
We'll wish you all the best in the next 10 years to come back with
thanks.
and hopefully you'll return to us sooner than that.
Yep.
I'd like to thank you again, Walter.
Thanks, Thor. Yep.